Here we are at chapter 13. Chapter 13 is kind of take chapter 12 and push it forth just a little bit. Um, we're moving on and we're talking a little bit more about the modern understanding of inheritance. Back in Mendel's day, they had no idea of what these things called chromosomes were or what DNA was or any of that stuff. They would just knew that there was some sort of information that was passed from parent to offspring in this thing called inheritance. Well, as we kind of progressed forth in the scientific pursuit, we discovered things called chromosomes. This was specifically kind of pioneered by these two gentlemen here, Sutton and Boveri, and the chromosome will be kind of discovered and observed in 1902, so just a few 20 or 30 or 40 years after some of Mendel's work. There's this thing called chromosomal theory, and that's basically what we now believe to be true about chromosomes. And I won't make you memorize all of these things. This is all stuff that we've already mostly talked about. But just FYI, in case you're interested to know, chromosomal theory says this, that there is a process known as meiosis, in which homologous chromosomes, meaning one from mom and one from dad, that are of the same size and bear the same genes, these homologous chromosomes will separate out into discrete structures that are independent of other chromosome pairs. Okay. Also, the way that a chromosome is sorted into a gamete appears to be random. So if here's the parent cell and here's some gametes that are being produced, and there's two chromosome number 7s, one chromosome number 7 could either go in here or it could go in there. The other chromosome will go into the other one. Okay. So it's pretty random which one chromosome goes into which gamete. Gametes contain half the number of the chromosomes in the somatic cells. So remember, a somatic cell is going to have 46 chromosomes. Gametes are only going to have 23 chromosomes. Both male and female gametes have the same number of chromosomes. That would be sperm and the ova, or ovum, eggs. And gametes combine to create cells of the same number of chromosomes as the parent. That's chromosomal theory. Before we talk about some of the disorders and inheritance issues here, we're going to want to bring up this word again called a karyotype. Now, a karyotype, what it is if you were to stain the DNA in somebody's, the nucleus of somebody's cell, that would be called a karyotype. And then often what's done is that the karyotype can be kind of photoshopped and arranged to form this thing, which is called a karyograph, which shows all of the chromosomes present and arranges them into the appropriate numbers. You have pairs 1 through 22. These are the autosomal chromosomes, and then there's a pair of sex chromosomes here. And it's important to keep this in mind because we're going to be talking about some genetic disorders that are inherited. And in order to kind of understand the severity of those disorders, you must kind of think a little bit about how much genetic material is on these chromosomes. So chromosome 1, these are going to be the tallest chromosomes. They've got the most genetic material on them. And then there's also a couple of chromosomes like chromosome 21 and 22 that are rather small. A real quick note here about genetic maps. Genetic maps are things that actually are going to tell where a trait is on a chromosome. And the way that these are made is actually pretty complex. But just in case you're interested, this isn't something that I'm going to require you to know on the exam. This is more of a genetics course level topic. But if you're interested, the way that they figure out where genes are on a chromosome is pretty sneaky. They look at recombination patterns. Okay, so basically, remember in meiosis 1, there's some crossing over, what's called. And in prophase 1 of meiosis 1, the two homologous pairs are side by side in this thing called the tetrad through a process known as synapsis. And then they're going to cross over and actually change DNA. Well, this whole idea about a genetic map is basically the further away traits are from one another, the more likely they are to be inherited differently. So, for example, like if the trait for antennae and the trait for eye color, this is in fruit flies, I think Drosophila. If the trait for how long the antennae is in these fruit flies or what color eye they have, if that's really far apart on the chromosome, it's likely that they could be one could be crossed over from the other. So, for example, like like imagine right here on the chromosome, this is for the antenna, and down here, this is the trait for eye color. Well, when they're really separated out like this, it's probably a pretty likely event that you know, the paternal, let's say this is the paternal chromosome, the one that was inherited from the dad, and then this is the one that was inherited from mom. Well, it could be that dad's genes actually get recombined, so that now suddenly on one chromosome there's dad's gene and then mom's gene. They've switched flopped. Okay, now if they do this frequently, where dad's gene gets swapped so that now mom and dad's gene are on the same chromosome, whereas they weren't before, when that happens frequently, that means that these are further apart. Whereas if you had two genes that were really close together, like let's say the wing structure and the eye color. So if, let's say, wing structure is right here and eye color is right here, these oftentimes are not going to be they're going to travel together. So if this crossing over event did occur, they would still be they would still be together, versus one on one chromosome and the other one on the other one. Okay, I know that's pretty complex. That's why you don't have to know that. But just in case you're interested, through tons and tons and tons of studies looking at the frequency of how traits are inherited and whether or not the two traits stayed on the same chromosome or they recombined into the other homologous pair of the chromosome of the homologous pair. You can kind of put together, through extensive studies, put together some of these charts to then figure out like where on a chromosome genes are. So you might figure out, oh, okay, this gene is here, this gene is there, this gene is here, and that gene is right there on the chromosome. And that's pretty cool because I mean that's pretty hard to do, is figure out you know, what genes are on a chromosome. Okay, now a little bit about inherited disorders. There's a lot of inherited disorders that have a chromosomal basis to them, and one of the bigger ones is this thing called non-disjunction. And what non-disjunction means is basically it's during meiosis, the chromosome will not separate correctly into the gametes. Okay, so there'll be sperm that have either too few or too little chromosomes, or eggs that have too few or too many chromosomes. And so here's kind of a picture of the process. Normally in meiosis, homologous pairs will line up side by side in meiosis 1 specifically. Okay, so here you have two cells that are each about to go through meiosis, and you can see that is in fact the case. Well, sometimes a non-disjunction event can occur during meiosis 1. If that happens, you can see rather than this being sorted out this way to create two new haploid cells, this homologous chromosome has accidentally gotten pulled into this place right here. And that would be the case if, let's say, these mitotic spindles didn't grab onto the kinetochore and pull it, and so as this spindle started to shorten, it took this one with it. And this is actually a good point. You can see why this is such an important part of the G2, the M checkpoint. We talked about the M checkpoint in the cell cycle. One of the features of that point is that the spindles are attached to every single kinetochore, because if they're not, you might get a non-disjunction event like this, and if you were to see what happens when that occurs, is it messes everything up. Okay, so if you look at all of the gametes that would be produced from here, every single last one has a chromosomal abnormality. Okay, well, non-disjunction can also happen even if meiosis 1 is okay. It can happen later in meiosis 2. So what you're seeing here is that these went to two separate new cells, these chromosomal pairs, and then these ones divided correctly. The two sister chromatids were pulled apart just fine, but here the two sister chromatids were not pulled apart. This one came off of that one, and that looked good, but you can see these two are still stuck together. This one should actually migrate to this side of the cell as well. Okay, and if you look at all of the consequences of these, if non-disjunction occurs in meiosis 1, it's a little bit more severe because every single one of the gametes has got a stranger number of chromosomes. There's going to be half the gametes that have a haploid number of chromosomes plus one additional kind of extra one, and then half of the chromosomes are going to have a haploid number of chromosomes, but then they're going to be missing just one chromosome. And then whereas if the non-disjunction event occurs in meiosis II, then it's a little bit less severe because half the gametes produced will still be okay, and then the other half are just going to be a little bit strange. There's going to be one extra chromosome in one cell and one missing on the other cell. Now, most of the time, and this can occur, and most of the time, what happens is if one of these gametes was the one that was used in the fertilization event, most of the time that developing embryo will not be viable. The embryo will not develop. It's just too much of a genetics abnormality for the embryo to survive. But there are a few disorders in which, for whatever reason, the non-disjunction event is not as severe, and then this can lead to the survival of the person. There just might be some symptoms or side effects to it. Now, the place where this is most often the case is when we're talking about abnormalities in the sex chromosomes. So there's a variety of disorders here that are involving the sex chromosomes, which may lead to the full-term pregnancy and you know, a pretty much healthy individual just with a couple of abnormalities. The first is Turner syndrome. Turner syndrome is where there's one chromosome missing. Okay, so if you were to like look at this picture again, this would be one of these scenarios where a gamete is made that's missing a chromosome, and specifically the chromosome that is missing is the X chromosome. Okay. So if non-disjunction happens and results in a gamete that's missing an X chromosome, then it could be the case that one ends up with Turner syndrome. Turner syndrome, they have one copy of the X and then they're missing a copy of the X. And this generally leads to basically a female anatomy. Female anatomy because the Y chromosome is the thing that's going to stimulate the developing zygote to procure male anatomy. So because there is no Y here, we know it's going to be female anatomy, but some of the hormones that are responsible for the maturation of female anatomy are on the X chromosome, and if there's only one copy of the X, it could lead to a hormone deficiency during puberty. So not as much of the female sex hormones, which are going to make some of the female anatomy a little bit more underdeveloped. And then one of the bigger side effects or consequences of this disorder is infertility. The production of ova, or eggs, is a pretty heavily hormone-dependent process, and so if there is a deficiency in the hormones, it's likely that the egg or the ova are not going to be produced as they ought to be, and that can sometimes lead to infertility. Okay, so that's Turner syndrome. Another example of kind of a non-disjunction event that happens with the sex chromosomes is one additional X chromosome, so 3 X chromosomes. Anytime there are 3 of a chromosome, we call it trisomy. So because this is an X chromosome, we call it trisomy X. And women that have trisomy X are going to usually be a little bit taller than normal, and they can sometimes have a higher rate of learning disorders. And I'm not exactly sure why that is the case or what specifically the learning disorders are, but trisomy X probably isn't as detrimental as some other non-disjunction things that could happen. Another one that could happen is one extra X, but when it's the male anatomy that's developing. So there is a Y here, X and Y, so that would be, this would differentiate into male anatomy, but then there's one additional X also here. So that's going to be just kind of leading to a little additional female sex hormone being produced, and it may result in a male that has either smaller testes or maybe even perhaps a little bit of breast development due to kind of the sex hormones being produced by that extra chromosome. And sometimes this is going to be so subtle that it goes unnoticed. Other times it could be a little bit more evident. Okay, the last one we'll talk about, these ones here are all dealing with X chromosomes. Okay. There is one that deals with an autosomal chromosome. There's actually, there's going to be, this by no means is an exhaustive list. There's lots of other ones here, but one of the more common ones is Down syndrome. Down syndrome is what it is. It's going to be trisomy 21. So there's a third copy of the 21st chromosome in that genome. And if you look, the 21st chromosome is this one right here, and you can see that the size of it is pretty small. And so that's why I think that trisomy 21 is a little bit more common, because generally if there's a trisomy or a chromosome lacking in any of these big chromosome pairs, then it's going to just lead to an embryo that's non-viable and it won't survive full-term pregnancy. But because the 21st chromosome doesn't have quite as much genetic material as the others, an individual that does have a third 21st chromosome can survive. And people with Down syndrome, it's kind of on a spectrum, and the severity of it can vary. But generally, some of the things that occur in Down syndrome is there's just going to be kind of a delay in development. And also, people with Down syndrome tend to have a harder time looking people in the eye, and even just a little bit harder time maybe communicating some of their thoughts and feelings. And so there's sometimes a little bit of a strange communication barrier that can be challenging for the person with Down syndrome. Down syndrome is a little bit more common. I think it's about 1 in 700 births about, and so most of us probably either know someone or have a sibling with or a family member with Down syndrome. There's a really great video here which I was able to find, and I won't play it here on the recorded lecture. I did play it, if you ask me, I'll play it in, I think I play it in lecture in class. But if you want to watch it, you can go ahead and copy this link. It's really great, not only because it talks a little bit about Down syndrome, but it also gives us a lot of helpful insights just about how to deal with people that have disabilities in general. I think that's sometimes a little bit of an uncomfortable thing to talk about, or it can be sometimes uncomfortable for people that have not a lot of exposure or encounters with people that have more, evident disabilities. It's a little bit hard to know how to act if you're somebody like that. And this is just a really helpful video kind of that shows a lot of insight and perspective into those scenarios. So go ahead and watch that if you have the time. I won't play it here in the recorded version of the lecture. One last thing about Down syndrome is that it's ends up being quite a bit more common in women who have kids later. So if a woman wants to have a child and she is older than 40 years old, that's kind of when it starts to peak. So by the time the woman is 45 years old, there's actually the risk of having a child with Down syndrome is about 3 or 4 times greater than if and then having a child in the 30s. Okay, so that's a little bit of thing to note there. All right, so there's some other chromosomal abnormalities that can result that are above and beyond just non-disjunction events where one chromosome is either missing or one extra chromosome is inherited. And these are the following different types of abnormalities that can happen. One is that a chromosome could just be duplicated, so there could be one region of the chromosome that's duplicated, not the entire chromosome duplicated, but maybe just a small region of it. Or one region of the chromosome could be deleted. An example of a disorder in which there's a deletion is a disorder called cri du chat. And cri du chat is the French word literally means cry of the cat. And that was coined because children with cri du chat, they have kind of more of a high-pitched cry throughout their younger years. And this is actually deletion of chromosome number 5. Again, there's another interesting video if you want to learn more about cri du chat. Again, I won't play it in this recorded version, but there's the link for it if you ever wish to watch more about it. Another thing could happen is inversions. Inversions are basically where a piece of the chromosome is going to break off. It's going to rotate 180 degrees and then reinsert back into its original chromosome. So if, like, let's say here's the chromosome and here's a little small chunk right here. Well, this could actually detach and then flip around so that this end is now over here, and then this whole thing can reinsert itself back into the chromosome, the original chromosome in which it came from. That would be called an inversion. Some of these can be more detrimental than others, depending on if the break happens right in the middle of a gene or not. If the break happens totally in between two genes or encompasses an entire gene but it's not halfway through a gene, then it's not going to be as likely to cause as many abnormalities. Then also another one that could happen is a translocation. This is where one piece from one chromosome is going to detach and attach to a piece of another chromosome. So here, this piece, rather than detaching and then flipping and reattaching back to the same chromosome, as would be the case in inversion, this one instead it breaks off and it's going to now attach to a different chromosome. And then this piece that was on this chromosome now gets flip-flopped over to here. So then you end up with these two scenarios where this little piece used to be here, but now it came over here. This big piece, which used to be over here, is now over here. So there's two chromosomes that just flip-flop some DNA. Now, keep in mind, this is not the same thing as crossing over. Crossing over is a very natural process that happens between homologous pairs. Two homologous pairs are going to have the same genes at the same locations, and they're going to be the same chromosome number. So this would be like chromosome number 1, and this would be chromosome number 1. Well, crossing over happens all the time, and it doesn't result in any sort of abnormalities. It just leads to genetic diversity. Well, translocation, because it's between two different chromosomes, let's say like chromosome number 1 and like maybe chromosome number 7 or something, that's a little bit more of a cause for concern. Again, if this break happens right in the middle of a gene, it's going to be more severe than if the break happened kind of in between two genes. But in either case, that's translocation. All right, that's going to wrap it up here for Chapter 13, nice and short and sweet. Here are some review questions to think about. What is non-disjunction and why can this lead to abnormal numbers of chromosomes? Give an example of a disorder caused by an abnormal number of chromosomes. And then give one more example of a disorder caused by a chromosomal abnormality. So this would be either a duplication, deletion, inversion, or translocation. Think about these and try and answer these, and that is going to wrap us up for Chapter 13.